I will just give you the introduction in this level, and then you'll go in batches. You see for yourself. See, one thing, what I'm telling you is that when you are on online platform, what is happening? You cannot question. No, you can question. No. Some thousands of students will question different questions. So in online platform, if you are studying with some centers, that is your personal choice. That I won't, because I don't. But you, with that input, I'm saying, not our input. With that input, you are seeing, visualizing in the lab. It can be the dissection hall, it can be in the dead body. Okay. And if you want to see dead body dissection and prosected parts, then you please, I told you, how many of you have taken Auckland's Atlas in your pen drive or hard disk? Are you finding easy to conceptualize? Because see, we are demonstrating from the in a hall, that big hall. Now over here, what I want to stress, whatever your teacher is telling you, that this is the humerus. These are two tubercles. This is the intertubercular sulcus. What structure passes through the sulcus? Now, in the holding the your humerus in an anatomical position, then the olecranon fossa, that is in the posterior aspect, and the head in the medial side, and it is being articulating with the glenoidal cavity of the humerus. This is said when you are seeing that video, that you see even the bones, they are telling you, oh, this is this. One model, young adolescent, not adolescent, just 18 to 20 years boy, he's in blue and he's showing you everything. What are the movements and everything. And when you are studying in your, after dinner in the hostel, be in a batch of 4 or 5, you have your computer, you have your book and atlas and also the bones. If you do it every day, every week, what is being taught to you, it will be, you will walk through the examination. I believe it. I'm not saying that you won't play games, you won't do fest, you want to go to Posh Mela, you do everything. But don't do everything like me. Anyway, I'm telling you, panic is one thing that is killing you. Panic. But you don't think of panic. Okay, another thing is that now today we'll be discussing these are the competency-based undergraduate curriculum. It is in the books. In your book is there, in your histology text is there, every book, after the preface and the forward and all this, then authors, identity, you are being shown what are the things. But A, N, 2, 9, and don't remember. That is for your logbook and your any reference in any khata, histology, or your lab book in physiology. That if the teacher says this is mandatory, you do it. But logbook, it is mandatory. Have you got your logbook from the thing? Anyway, you will be getting it. Okay. Now, what I want to say is that now, the small intestine, general features of the small intestine, cells of the small intestine, duodenum, jejunum, ileum, Peyer's patch, large intestine. We will be pertaining to this up to parts. This. Okay. Small intestine. The small intestine is a 6-meter long mucomuscular tube. The small intestine has 3 parts: duodenum, jejunum, ileum. 6 meters. Now, it is a mucomuscular organ. So, your thought comes, why mucus? Why muscular? Because it is propulsing by peristalsis, and by it goes through from the oral cavity. Your food goes down the oropharynx, then it goes into the esophagus, then goes into the stomach, it goes into the duodenum, and then goes into the jejunum, that goes into the ileum. Ileum again is the ileocecal valve. Just below it is the appendix, and that cecum is there, and the large is ascending colon, transverse colon, descending colon, pelvic colon, and rectum and anal canal. This is the pathway. This all the process of digestion and this thing, it goes down with the enzymes and different. But mucus has to be liberated to carry. Second thing, propulsion is carried by circular and longitudinal muscles of the definitive organs. Now, the general features of small intestine is a site of digestion and absorption of nutrients. For sufficient digestion and absorption, small intestine has following modification and increases surface area. Plicae Circulares, circular folds or valves of Kerckring, villi, microvilli, and scripts of liver cone. Now, when you see this, your viscera, now see, this is the mesentery. These are the plicae circulares, valves of Kerckring and small intestine. This is small intestine. Now, segment of the small intestine shows plicae circulares. This is a cut open, that cut open. Naturally, the inert thing is that it is like Bengali Babu. If you see any Bengali film of Uttam Kumar or any Soumitra or anything, or Bengali Babu's typical dress is a kurta, not nowadays haldi kurta or red kurta, it is not that. It is a white, and the thing, the sleeves is, when they are ironing, you give it to the dhobi. They actually have a technique. They give a starch to the thing, and then they have a fruit. It's a square fruit. They catch it, and they go on like this. And that gives a nice look. Now it is like that. So the surface area is increased. This why this is now this your if it is like this straight, then 6 meters would have become 8 meters. So that 8 meters will be straight. But what is happening? It is having inner folds like this. Now this fold is like this. This is like this. Now see, if this is straightened, then it will come like this. So that thing is done with the mucous membrane in the intestine, within the intestine, to give you the surface areas. To increase the surface area, so if the food is going slowly, that gets time, the enzymes are being laid, and that gives a quicker absorption. Anything goes wrong, say it goes wrong in acute gastroenteritis. If there is inflammation of the mucosa due to some food poisoning or some pathological any E. coli infection or anything, then what happens? There is erosion. So whatever food you are taking, you go for, of course, in your age, definitely. I like spicy foods. I had craving for many food, but that you had that power of digestion. Natural, no, kebab order कर दिया, ये order कर दिया, इतना dinner में. Anyway, you have to enjoy. Don't think of gastroenteritis. Okay, and you take a PPI or some antispasmodic if you feel that you will be enjoying the dinner rather than think of this thing. So your Plicae Circulares, valves of Kerckring. Now it can come as a MCQ. Valves of Kerckring. Just one this thing. And now this is a villi of small intestine. See the villi, the mucosal surface of the intestine. This is the mucosal surface. Openings of the crypts. This is the epithelium, lamina propria, and this is the mucosa. The muscular is mucosa and submucosa. These glands are there, crypts are there. This is the design made by the Creator. If you believe in God, it's God. If you believe in Allah, it is Allah. Whatever it is, you believe in Christ, it's Christ. So it is designed in such a way with the evolution. If you go into comparative anatomy, then see elephant, such a giraffe. These, if you go into the comparative anatomy, then there will be some differences because of the type of food they take. Okay. Now, now cells of the small intestine. Now the intestine is lined by simple columnar epithelium. This lining epithelium consists of 5 types of cells: enterocytes, goblet cells, Paneth cells, enteroendocrine cells, and M cells. Now, enterocytes, absorptive columnar cells. Now, this will go and see how much we can in a light microscope without any special stain, with only H and E stain. Then you just have an idea what the gut is. Even with this, when I was a house staff in Bardwan Medical College, we came across a man with intestinal obstruction. Intestinal obstruction can be of many types, but this, when we went for laparotomy, now what we made up, our teacher, surgeon, professor, who made an incision, a big incision, okay, and opened the thing. Then what we saw, a major part out of 6 meters, more than 2 meters or so, it was gangrenous, black changes. So, sir clamped the healthy portions and slowly removed the 2 meter of the intestine. Now, this was an interesting case. At least I have opened how many, I don't know, abdomen in my career. I have seen many in my house staffship and internship, and but I have not seen such a case. and the total management I had to do. What food, what this thing, whatever, the post-operative management. And I took it as a challenge, and I really, that patient got well and he was discharged without anything. Of course, there will be a portion is being removed, 2 meters portion is removed. Now there will be some like or any fat food or something, then there is a type of diarrhea or some this thing. We were cautious, and we told them, advised accordingly. Now, this is I'm telling you an experience where I encountered 6 meters. Now you can do gut-to-gut anastomosis, intestinal anastomosis, or bullet injury or something like that, or one ballam, one spear, or blunt injury, like you are in the practicing in the gym. Okay, it gets contusion. There will be hemorrhagic areas. But this was a big experience we had, and I still remember. Now, goblet cells. Now, it is like shaped like a drinking glass, and broad above and narrow stem towards basement membrane. Now, drinking glass, whiskey or some cocktail, it is like that. I think I need not draw a diagram for that. Okay. You think of picture of Modi drinking apple juice and the Italian lady, she is drinking whiskey. And they are just toasting it. Okay. Now, Paneth cells, enteroendocrine or argentine cells present among the lining epithelium of the intestine. Cells produce different peptide hormones such as enkephalins, vasoactive inhibitory product. Silver staining, these cells can be stained by silver salt. This is a special stain. There are two types of enteroendocrine cells. Now, enteroendocrine cells is having a big role. Now, that you please read further. Now, cells of the small intestine, M cells, Peyer patches. These are epithelial cells overlie the Peyer patches and either intestinal lymphatic follicle. M cells, deep pocket like recesses. M cells, microfolds. Okay, antigens transporting cells. Now, M cells act as an antigen transporting, intermediate and undifferentiated. Okay. Now, this is stomach. Okay, this is the cardiac end. This is the pylorus. It is more muscular and quite hard because it contracts like this, pass the food. Okay, then you can see the rugosity, the folding. To increase this, you see your empty stomach. You had no breakfast. Now you go to the dining hall and you say, chicken hai to chicken, Thursday chicken, shag and all this. Now that goes inside. And if you belch, Descartes, that means you had a good excess of food. Okay, now what happens? This is the muscle, the serous layer is there. You will see adventitia. That when you take a section of this, you will be seeing it under the microscope with all these layers. Okay, now from jejunum it comes, the rest is the small intestine, ileum. Now this end, see, it opens into the cecum when you will see in the dead body. But I'm just telling you, see, it is the plica. Get it? Get it? This is the plica. It is like, ওটা বাংলা কথা হচ্ছে গিলে 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 করা পাঞ্জাবি. Okay. Now, okay. Now this, what I told you on the previous slides, that it is like this. It increases. Over there I drew the picture. Okay. Now please go and start seeing. Have any problem, you ask any of the teachers in the faculty. I'm not his batch or her batch or this is that, doesn't matter. You ask if you have any problem, ask. Second thing, you 60% you do it yourself and your lab and your dissection hall, your study room and this thing. And 40% if you want to do, if you feel easy, go for online. But I would say that that's up to you, your personal choice. But don't go for online for this thing. Everything you think that is, it will be done. It will be done. No. Today you see, immediately you see in the lab, you see in the your textbook, khata, drawing, rough sketch you do, and see and label. Whatever you see, label. You have to, you have all the potentialities. You have to generate. You have to, it's a habit forming. Like early morning you're going to potty. It's a habit. Some people smoke before going to potty. I don't know if your friends are smoking. Anyway, so you have to make it a habit. Ekdom. So this you see, correlate, and after 10 p.m. you go for your batch to not single. Study together. It's a teamwork. So you have to, you just don't keep silent. Hey, tu bhul korchis. Dhar, bhalo kore dhar. Like you are a ustad, but you are in the same level. Understand? You correct. No. Head will be on the medial side. The olecranon fossa will be in the posterior side. That's the anatomical position. So similarly, you won't be having the specimen there, but you're having the specimen in the lab. আজকে দেখিয়ে দিলাম, আবার তোমাদের গ্রস অ্যানাটমি হবে. You go and see. কেউ আটকাবে না. Dead body পড়ে আছে, তুমি দেখছো, but don't tamper, don't waste, because it's very difficult to get specimens. In Hunterian Museum, built by John Hunter, you please search your Google, who was John Hunter? Okay. Now you see his photo in the dissection hall, in the end, it is there. Now if you study bit of history, why I put it there, you just study, just go through, like you are reading one novel, or Agatha Christie, now please go. Now I think I have to go there. I will show you. You just don't panic, and you see the stomach, the rugosity. It's like, if you flatten it, when it comes flattened, bloating, acha khana khaya. In Facebook they show, itna bada thala, so it is like that. Now that goes, it increases the surface. But that has to be accommodated in your stomach, in your body cavity. Okay? Now please, you want to see. Okay, and you see, this is the adventitia. You go like this, view, and slowly see all these layers. And this is longitudinal muscle. If it is like this, longitudinal muscle fibers. Now this, if it is cut like this, transverse section, this is a transverse section. Okay, yes, then what happens? The fibers will be like this. And the circular muscles is like this. It is in this. You just see the total design of this slide. And this is the best stained among these three slides. So please view. And now it's not the exams, so you can see I from adventitia. This is the external coat. Then circular muscle, longitudinal muscle. Then the mucosal lining, it won't be flat, I told you. It will be like this. And over here, you see blood vessels, artery. This is one vein. Over here, packed with RBCs, See? Chalo. তারপরে দেখে নিচ্ছি and you just up and down you from the adventitia, the circular muscles, longitudinal muscles, submucosa, all vessels are there, artery. দেখলে তো artery-টা, and don't make this thing that this is Peyer's patch. No, you can see the small Peyer's patch is there, but artery. It's just beautiful. This slide is. Now circular packed with RBCs are there. দেখো আমি তো বলছি হ্যাঁ আর্টারি ওই তো দেখাচ্ছি এটা পায়াস প্যাচ দেখছিস তো ডেন্স dense, but see this is this thing, and I will show you. देखिए बाबू एक मिनट. Now you see over here there's a big artery over here with packed with RBCs, and here it is bit distorted, but you can see the Peyer's patch. আর এখানে আর্টারিতে রেড দেখ রেড কাপাসুস আছে, you can see the arterial wall even. See muscle layer, adventitia and everything. দেখো না, দেখো এক এক করে দেখো, ওখানেও দেখতে হবে ওই দুটো it is light stained, মানে it is predominance of eosin, মানে হেমাটক্সিলিন ইওসিনে যেটি আছে, হেমাটক্সিলিন যখন it is a water based this thing, তো সেখানে ওই একটা প্রসেসিংয়ে কোথাও ইওসিনটা মানে হেমাটক্সিলিন ইওসিন নিউক্লিয়াস স্টেনটা সেভাবে নেয়নি. That can be that's a problem with the technological problem. That's why if you একই সেকশনে কিন্তু one tissue, one block from same section is taken. If you see the topography of this slide, that slide, it is from the same block, but it is beautifully stained. The eosin component, hematoxylin component is beautifully stained. Over there, you see it is the light nuclear stain is not so predominant, but you can identify the nucleus. But there's a difference between the three slides. So those slides won't be given. This will be given if it is given in an exam. We will give the best slides, which is having very good stained slides. It is not to put you in a trap or lang nahi marega. लाठी दे देगा आपको, ঠিক আছে লাঠি দিয়ে দেবো কিন্তু ল্যাং মারবো না. No one is here to what I want to, my personal feeling is that this is the only place you will learn, not place means anatomy, I'm telling you. After that you will forget. But day to day you have to come to the anatomy reference. That's why I've told you that's a gateway to medicine. if you read a report, left ventricular hypertrophy, ठीक है जी? And or you see the see your a radiograph, it gives a big heart. That might have had a hypertensive, all this thing. So his ventricles were hypertrophied.